Hello?
Hi.
Oh. Can you put the volume louder, or do you have...
Louder. Okay.
Settings.
Is it me?
No, not you. You can't now.
Uh-oh.
Oh, oh, if he's just stopped sharing screen, that's right. This is fine. And basically, the volume, it should be.
Can you hear me now?
No.
Hello?
Hello. Can you hear me now?
Pretty quiet, isn't it?
Yeah. I can hear you, but it's quite... It's a bit quiet. I did. It's the same volume.
Okay, I'll see if I can increase my volume.
If you can, it'd be great if you could.
Is that any better?
Sorry?
Is that any better?
Yeah, that's better.
Might be a bit of headphones.
Okay.
Yeah.
Are we good to go?
Yeah. Have you screen shared it?
Yeah.
Okay, great.
Again.
Great.
Brilliant. Have fun, guys.
Yeah.
Excellent. Okay. All right. So we're starting with this this paper too. I believe it's winter 2017.
Yep.
So have a read of that and let me know what you think.
Okay. Okay.
Okay. So talk me through how you do this.
Okay. So the formula is M equals I divided by A.
Yeah. So M equals I over A. Can you see me writing there?
Yes, I can. Yeah. The magnification we already know is times 16,000.
Mm-hmm.
And we would get I by measuring the line XY.
Okay, I'm going to tell you that I equals 40 millimeters.
40 millimeters, okay. And that in micrometers is 40,000 micrometers.
Okay, so you have to stay here. Because Joyce has now
I can move.
Thank you.
Yeah. Okay, yeah, so 40,000 micrometers. And then I would divide 40,000 by 16,000, which is the magnification.
Yeah.
And I would get the actual size.
Okay, good. So
Yeah.
that's correct. 40,000 divided by 16,000, which gives you 2.5. And what's the units there?
The units is it's micrometers.
Yeah. Perfect. That way you get all the marks.
Yeah.
So the next question is the organelle A and state its role. So just take a quick look back at this.
Okay. Mitochondria.
Okay, good. That's one mark.
Aerobic respiration. So ATP synthesis.
Yeah, perfect. So that'll get you the mark. Moving on, name cell structure will be in state one reason for your answer. So
The matrix. Or is that touching the cristae? I can't.
so B is actually outside
Oh.
of the mitochondria.
Oh, B. Sorry. Okay. B. Okay. That's the rough endoplasmic reticulum.
Okay, and why are you saying that?
Because you can see the membranous sacs, and you can see the dots which represent ribosomes.
Yeah, perfect. So the key here
Is the ribosomes.
Yeah, exactly. So without an explanation about just shorthanding there. So if you didn't see ribosomes, what organelle or organelles could that be?
If I didn't see the ribosomes, I could have thought it would be Golgi apparatus, maybe, or smooth as endoplasmic reticulum.
Yeah, perfect. Good job. Let's move on to the next question. Right.
Okay. Because the left ventricle has to pump blood throughout the body, while the right ventricle has to pump it towards the lungs. Thus, more pressure has to be created because it's a further distance for the blood to travel.
Yeah, okay. So you get the marks for that. It might be simpler just to sort of reduce your response to
Okay.
points. So, the first reason why the wall is thicker in the left ventricle is because it has to generate higher pressure. So, as you say, I mean, you got to that point, but you first explained the different circulations. So, if I was answering this question, I would start off by saying that it's to generate a higher pressure, and then go on to say because more pressure is required to overcome the higher resistance of the systemic circulation.
Yeah.
Okay. And then you can confer that you can relate the distance that it has to travel as you did. And you can refer to the right ventricle generates a lower pressure, damaging the capillaries of the lungs, because if it matched the pressure of the left ventricle, you'd be pumping blood with a much greater force through a much smaller space, if that makes sense.
Yeah.
Okay. All right, just take a look at this for a second, and then we'll read the question.
Okay.
So first of all, name blood vessels F and G.
Okay. G is the pulmonary artery.
Yeah.
And F is the vena cava.
Yeah. So
Or just vena cava.
yeah, it's a vena cava. Which vena cava? So you actually have two vena cavas.
Okay.
F is pointing to the inferior.
Yeah.
If there was another arrow here, it would be the superior.
Okay.
Just go back down and see what the next question. Okay, name the stage of the cardiac cycle at K.
Atrial systole.
Why do you say that?
Because the blood is going from the atria into the. Oh no. Okay. It's diastole.
Yeah, this is diastole. Okay, talk
Yeah. Because so the blood is going into the atria, and some of it is going into the ventricles because the pressure has been relieved in the atria. So the semilunar valves are open too.
Yeah, good. This is diastole, and that's a correct explanation. So next question, describe what happens in the heart between stages H and J. So if we go back up, this is a 5-mark question.
Okay.
So, one give me 5 points on what's happening between H and J.
Okay.
Do you know what process they're asking you to describe here?
Sorry.
Do you know what process you're being asked to describe?
The process in between the atrial systole and the ventricular systole. So you're describing how the electrical signal passes through, really?
Yeah, so that's the first part. And also what's happening in J is you're getting blood leaving the ventricles. So
Yeah.
but you're on the right track. So go on then, explain to me the electrical side of what's happening here.
Okay. So the electrical excitation wave produced by the SAN can't go directly from the atria into the ventricles because they're fibers that they're fibers that don't allow the signal to pass through. So it goes through the AVN, where it's stopped for about 0.1 seconds, and it's passed on to the Purkinje fibers down the septum and up the ventricles.
Okay.
And the cell muscles, the cardiac cell muscles of the ventricles respond to the electrical excitation by contracting.
Correct.
So they contract. So the blood flows from the bottom of the ventricle. Not the blood. The electrical excitation wave goes from the bottom to the top of the ventricle, thus allowing the ventricle to pump the blood towards the arteries, opening the semilunar valves because of the blood pressure created by the muscle wall contracting and pushing the blood into the arteries.
Good. That's correct. In terms of talking about the bottom and the top of the heart,
Yeah.
it's we kind of use a different terminology.
Okay.
Can you think of any other words to describe what you're seeing here as the bottom of the heart?
No.
We talk about the base and the apex.
Okay. The base.
Yeah.
Okay.
So the base is kind of illustrated. I don't know if you can see my cursor.
Yeah, I can.
Yeah, so it's sort of like the back wall of the heart is the base, and the apex is here. So
Okay.
the electrical conduction will go from the apex towards the base.
Okay.
Okay.
So it goes from the apex to the base, and then from the base to the apex.
Yeah, I mean it's what's important to know is that the current is basically moving upwards, causing the ventricles to contract rhythmically in a sort of upwards fashion.
Okay. Yeah.
You can also say at the end that the atrium relaxing as the
Yeah.
next step.
Which allows the blood to flow into the atria. Yeah.
Exactly.
And fill them up. Yeah. Okay.
Okay. All right. Next question.
Okay.
Happy with that?
Mm. Okay, so alveolar type 1 cell is a cell with a thin cytoplasm, which allows the alveoli to have a thin wall, a single cell thin wall
Yeah.
for diffusion of respiratory gases to occur at a higher rate.
Yeah, good, exactly. So you get one mark there for saying that the cell is thin.
Yeah.
You get a flat or that it's squamous. And that adapts it to its function because it allows it to diffuse across a short versus the amount of diffusion that occurs.
Okay. Okay. So phospholipids have a hydrophilic phospholipid head and a hydrophobic tail.
Yeah.
So the hydrophilic phospholipid heads are attracted to the water, hence hydrophilic, and form hydrogen bonds with the water, while the tails, which are hydrophobic, so not attracted to water, are not don't form hydrogen bonds with the water and remain insoluble.
Yeah, good. And if you wanted to actually look at, we zoomed in on an alveolus, and this was surface of the membrane, and here was the lumen,
Yeah.
and the surfactant might look something like this, etc., etc. So these hydrophilic heads are going to be forming hydrogen bonds with water, whilst the hydrophobic tails sort of compact and align with the membrane.
Yeah.
Okay. Perfect.
Okay. Macrophages. Okay. Macrophages are white blood cells and phagocytes. So they engulf invasive microorganisms and dead cells
Yeah.
by phagocytosis, where they the lysosomes in the macrophages fuse together with the whatever is engulfed, and the digestive enzymes of the lysosomes destroy or break down the engulfed material.
Yeah, okay. That's
So
good.
they fight off infection diseases.
Important part. So remember, the questions asked for the role of the macrophages.
Yeah, not how. Yeah.
I mean, you'd still get, so you get 1 mark for mentioning phagocytosis,
Okay.
and you get another mark for mentioning that basically what phagocytosis is, so engulfing,
Okay.
destroying. But you could actually get all of these marks without going on to talk about that. So you got the third mark at the end by saying that it prevents infection.
Yeah.
You could, for instance, have named an infection in the context of the lungs, for instance, TB or pneumonia, and points relevant to the question, preventing infection at the stage of the lungs furthermore prevents the pathogen from entering the rest of the body.
Okay.
So you would get another, about that.
Okay.
And yeah, you could also get a point for talking about how macrophages are antigen presenting cells and how they activate the adaptive immune system.
Yeah, I was thinking about talking about yeah. Okay. Oh.
All right.
Okay. So elastase are so they secrete elastase because they're enzymes which break down the elastin fibers of the alveoli, thus allowing the neutrophils to enter the airways and fight off the infection.
Good. Yeah, that's both marks. So one mark there for saying describing the action of the enzyme to break
Yeah.
the elastin fibers, and another mark for saying that that creates a pathway for the neutrophil to enter the alveolus.
Yeah. Okay. Ooh. Okay. So by producing this protein, they don't have the inhibitors to prohibit the action of elastase. Thus, more of the elastin fibers of the alveoli will be broken down, leading to the alveoli not being able to recoil and expand, thus making expiration and inspiration more difficult, which also can result in the bronchioles collapsing and the air being trapped in the alveoli, thus the alveoli bursting, which decreases the amount of exchange of gases and can cause emphysema.
Yeah, correct. Perfect. So that's a really thorough explanation. You get all 3 marks there.
Yeah.
Good. Okay, so that's U25. Shall we take a 5-minute break?
Yeah, that's totally good. Yeah, 5-minute break. Hello?
All right, so let's pick it up.
Sorry?
So sorry, I was just saying, let's pick it up.
Okay. State the name of the pathogen that causes malaria. Plasmodium?
Yeah, correct. And the specific species, and so Plasmodium is the family. And then
So malaria or what?
yeah, that's what I meant. So Malaria. You could also say Oak Valley, Vivax,
Mm-hmm.
or most common killer. But yeah, that's that. Okay.
Okay. Female Anopheles mosquito.
Yep. Correct. Read it,
Okay. I don't. Okay. Mm. Okay. Mm-hmm. I'm just looking through the table. So.
Yeah. So 3 marks.
Yes. Numbers of cases. Okay. Numbers of death. Okay. So trends. So the numbers of cases of malaria in the African region is always smaller. No. I'm sorry. That's okay. The numbers of cases of malaria in the African region has decreased from year 1998 to 2013.
Yep. Correct.
So has the number of deaths of malaria from malaria in the African region.
Yep.
The number of cases in all countries has also decreased. And also the numbers of deaths in all of the countries has decreased.
Good. That's 1 mark. So in this one, they'll give you a mark for just describing that the numbers of cases have decreased in all countries or Africa,
Yeah.
or that the number of deaths have decreased in all countries or Africa.
Okay.
The other 2 marks, you need to comment on the other data.
So the number of cases in the African region is always smaller than the number of cases in all countries, which means that malaria is also present in other countries other than the African region.
Okay, yeah, that's correct. But if you wanted to describe the other trends that you can see, so from 1998 to 2013, there's also a change or a lack of change in
Okay, so the cases and deaths in Africa as a percentage of all countries the percentage of deaths in Africa as a percentage of all of the countries stays relatively the same.
Yeah.
throughout, so from 1998 to 2013, and there is a generally the cases in the African region as a percentage of all countries stays the same generally. So from 2003 to 2013 it does.
Yeah.
But there's a big dip from 1998 to 2003, but
Good.
they asked for the trend, so
Yeah, okay, so you'd get all the marks for that description. For that final bit, I would just say from 1998 to 2003, there was a large drop in cases, and then it
Okay.
plateaued with minor fluctuations,
okay.
something like that.
Okay. Was there anything else that I could have said there?
No, you picked up all the marks. So they essentially wanted you to just explain what's happening in each column.
Yeah.
So, you could just write 6 sentences, and that would pick up all the marks.
Okay. So one, because of the development of the vaccine against malaria, has been become more successful over the years. Second point would be that it's easier to vaccinate people because of the dipstick test.
Okay.
Yeah, that's all that I've got.
Okay, so talking about the use of new drugs to prevent transmission or prevent
Oh.
or treat malaria, that's one mark. Okay, the other 2 marks you have to talk about other things that have happened between 1998 and 2013. So other than drugs,
Yeah.
think of anything that if I was to ask you.
So maybe the increasing of awareness of preventing the mosquito from biting people by mosquito nets and insecticides.
Good, yeah, you get a mark for that.
Maybe yeah. I don't know if this is a point, but the drainage of ponds and stocking, maybe. But
Okay, yeah, you'd get a mark for that. Tell me why.
okay, so by draining and stocking ponds and yeah, so you decrease the number of mosquitoes, thus the numbers of vectors, decreasing the number of infected people.
Yeah, good. So you're essentially controlling the breeding of mosquitoes or the population size.
Yeah.
Drainage of stagnant water counts as one of those methods, so you definitely get a mark for that. Bed nets and insect repellents and insecticides come under the broad heading of reducing contact between the vector and humans. Mentioning that. And then, yeah, you get another point for talking about the use of new drugs to prevent or treat malaria. You could also talk about earlier identification of cases. So as our knowledge about the disease increased, we were able to figure out which signs and symptoms happen earlier on in the disease, and that led to people being treated more.
Okay.
And another point is that through awareness and further education about malaria, the disease, the transmission, and the control methods, we were also able to decrease the number of cases and deaths. But they're kind of like those final points are a bit superfluous. I think the three key points here are managing vector, managing its contact with humans, and developing new drugs and all those points. So that's good.
Okay. So malaria is very difficult to control because it's caused by Plasmodium, which mutates. So it has different proteins at different stages of the transmission cycle. So either vaccine would have to be made against all of the stages or just the infected stage.
Okay, good. That's one point.
Okay. Malaria is also difficult to control because there's a vector involved.
Yep.
With
So
the transmission. Sorry?
Tell me about why it's difficult because there's a vector.
Okay, so it's not direct transmission, so it's harder to control the transmission cycle because it's harder to, you have to get rid of the vectors. That makes sense.
Yeah, if you wanted to completely eliminate it, that's correct.
Okay, and malaria is also hard to control because there's an increasing so the climate change is making the conditions for the mosquitoes easier to survive in. So the conditions are always getting better for the mosquitoes, for the vectors.
Yeah.
So also because of the difficulties that we are having creating the vaccine, the appropriate vaccine. And
So you've covered good points in terms of developing the vaccine and the fact that you have this vector pool that can breed like crazy under the conditions which it's in. Other reasons why malaria is difficult to control, you take a more social perspective. Can you think of any ways that it's difficult to control because of the sort of human aspect of controlling a disease?
because it's hard to know whether or not, or it's hard to prevent the mosquitoes from infecting you, from biting you.
Yeah, it's also just it's difficult to identify people who are infected.
Yeah, because they don't have any visual symptoms.
Yeah, and especially if the people who are exposed to infected individuals aren't educated to be able to spot the signs of infection.
Okay.
There are other, like, broad topics that you can touch on. For instance, the regions where malaria is a problem, often there's inaccessibility to healthcare, which makes it difficult to control because people aren't treated once more. Also, in these regions, there are a lot of people who are infected with HIV, and AIDS predisposes people to a greater risk of fatality when infected. And problem in a lot of these countries is to implement a malaria control program, you need resources, and a lot of the governments either don't have a lot of resources to pay for the drugs or insecticides or vaccines, or they don't have the resources to fund research. So you can talk about all of those things as well as the sort of biological mechanisms that make it difficult to control.
Okay.
All right, let's move on.
Okay.
That makes sense.
Yeah. Okay. Okay. So transpiration is the diffusion of water vapor from leaves down the concentration gradient to its environment from air spaces where the water has been evaporated into.
Good. So you get 1 point there for talking about transpiration being the loss of water vapor, and another point for saying that it leaves the leaves of the plant, the aerial parts of the plant.
Okay.
Okay, so for this next part, I'm not going to ask you to quote the specific values because it's kind of difficult for us to talk through it when I'm not there. But if you could just describe the changes, the mean water potential, and then suggest the explanations. So,
Okay, so there's a decrease of the water potential until about 2, and then there's an increase of the water potential.
good.
That's a trend. And the explanation is tied into the rate of transpiration during the day. So, okay. So the rate of transpiration increases and then decreases, but there's also a time difference between when the water potential decreases and there's a high rate of transpiration. So by there being a high rate of transpiration, more water is diffusing out of the leaf cells.
Yeah.
Thus, the water potential decreases when transpiration increases.
Yeah.
Yeah.
Okay, and in terms of what's actually happening in the plant, so talk about the synthesis of solutes, talk about xylem, talk about stomata. Can you add some detail to that?
Yeah. So, so transpiration creates a gradient which water can flow down by. So by water leaving the plant, it occurs. It makes the water evaporate into the air spaces from the wet cell walls of the mesophile cells, thus causing the water to move into the mesophile cells, either symplastic or apoplastic pathways, which then causes the xylem, the water to flow up the xylem down the concentration gradient set by transpiration.
Yeah, and why does it sort of increase during the day then?
Why does
Why does transpiration increase?
Because of the light intensity, that's why it increases and then decreases during the day. So as there's a higher light intensity during the day than during the night. More stomata are open because photosynthesis can occur, and CO2 is also diffuses into the cell as well as transpiration occurring because the stomata are open.
Correct. So that's the key point. There's stomata opening because basically it's not, it's a response to light intensity as photosynthesis occurs under high light intensity. That tells the plant that it's not. As soon as they open, water vapor starts leaving the plant, and that increase in transpiration. But yeah, that's good. You covered the points there. So let's move on.
Okay, reduce the water potential gradient, water vapor between air spaces. So it leaves the screen. Yeah, okay. So this can be done by having hairs on the surface of the leaves, thus trapping the water vapor which diffuses out of the plant through the stomata and decreasing the water potential gradient of the between the air spaces and its environment.
Yeah. Okay.
So that's one. Second one is by rolling the leaf, so thus creating a more humid environment by having the upper. So on the upper epidermis, they would have stomata, and they would curve the leaf so that there's a more humid space in the middle of the leaf, which decreases the water potential gradient between the leaf and its environment.
Yeah.
Those are two. Another one. So, no, a waxy cuticle wouldn't.
So they want you to talk about sunken stomata.
Sunken stomata, yeah. Okay.
So tell me about that.
So sunken stomata are stomata which are inside the leaf itself. So, thus being closer to, thus decreasing the concentration gradient, because when water diffuses out of the stomata, it's still near the leaf on
Yeah.
all of the sides.
Yeah, so sunken stomata, the thing that it does is it sort of protects the water vapor from air currents.
Yeah. Oh, okay.
Have like this here, and normally stomata would be like this.
Yeah.
Then where it escapes air quickly. But
Okay.
instead, you have something like this with
Thus, yeah, wind
Right.
doesn't affect.
yeah, the wind sort of affects this water vapor less.
Yeah.
Does that make sense?
Yeah.
Okay, perfect. So that's the three points they want you to talk about are sunken stomata, trichomes, or hairs, and the fact that the leaves are curled.
Okay. Involved in this control cell cycles and okay.
So two more questions.
Can you scroll up quickly? Okay, great. So EGF is a signal which binds to a complementary with a receptor with a complementary shape,
Good.
which cell A has, while cell B has receptors, but they don't have a complementary shape to EGF, thus can't bind to it.
Good.
And
That's all you
no
have.
response. Okay. More RNA is produced than because more proteins need to be synthesized.
Yeah.
DNA is okay. So free nucleotides are used, so they bond with complementary bases by hydrogen bonding.
Good.
So they're used during the S phase for DNA synthesis, and DNA polymerase, which catalyzes the bond between sugars, the pentose sugars, and phosphate groups,
Good.
forming the backbone of the molecule, is used during the S phase. And the phosphate groups and the sugar groups themselves are used to form the backbone.
So they're part of the nucleotides. If you have free nucleotides, they have a sugar and phosphate group. So,
Yeah.
Yeah, you get a mark for nucleotides, you get a mark for DNA polymerase. To get the last mark, you have to talk about something else.
Okay. And
So you mentioned DNA polymerase. That's an enzyme that's involved in sort of rebuilding the chain. Can you think of any other enzymes that are needed?
oh yeah. So to unzip and unwind the DNA molecule, helicase
Yeah, good.
is needed.
You get a mark for that. You could also talk about topoisomerase. Have you heard of that? So it's an enzyme that's used to sort of change the superstructure of the DNA, so change the coiling to allow all these other enzymes, nucleotides to come in to actually allow DNA to be synthesized.
Okay.
We also talk about DNA ligase, which has a role here, and ATP, because this is an energy-driven process that requires ATP.
Okay.
Just do the last question for the break.
Centromere. Oh no, wait. Yeah,
Yeah.
centromere.
Good.
Function is the position of the centromere determines what type of chromosome it is. Added to that, it also contains cytokines which allow microtubules to bond with the centromere, allowing it to split up during the anaphase.
Yeah. So kinetochore.
Sorry?
So you said cytokines.
Yeah.
Do you mean kinetochores?
Kinetochores. Sorry.
Yeah.
Cytokines are used in the immune system. Yeah. Yeah. I mean kinetochores.
Okay. And then the last one. Right. You don't need to draw. Just
Yeah.
explain to me.
Okay. So I would still. So I would draw the two sister chromatids being pulled towards either both of the poles. So the I don't know how to just so the legs or the ends of the sister chromatids would be closer to the middle than the centromere, which is being pulled towards the poles.
Yep.
And I would show the mitotic spindle being attached to the kinetochores and the centrosomes.
Good. You get the marks for that. So they just, if you were drawing this, they'd want you to definitely make these two identical.
Yeah.
You couldn't draw more than one because they're asking you to only draw the chromosome.
So you can't draw more than one.
Yeah, they just want you to show that chromosome.
Yeah.
And if you look closely, one of the legs is clearly longer than the other.
Yeah.
So that detail would get you a mark as well.
Yeah.
And the V-shaped chromatids with the centromeres pointing towards the poles gets you a mark as well. So you get both marks there. Okay, that's good. Let's take another 5-minute break, then we'll pick up on the next pathway.
Okay. Hello.
Hi.
Are we going to another paper?
Yeah. Are you happy with that?
Sorry?
Are you happy with that?
Yeah, that's fine. Okay. Closed double circulation. So, double because the blood passes through the heart twice, and closed, because the blood is transported in closed vesicles.
Yeah, correct. That's both marks. You said that the blood passes through the heart twice. What do you mean by
In one cycle. So it passes in the systemic and the pulmonary cycle.
good? Yeah, so there are two different circulations. Let's take a second.
Okay. Okay. Pulmonary vein. Okay. Then left ventricle, then
What are you doing?
aorta,
Oh,
then aorta, then vena cava, then right atrium.
Yeah, perfect.
Okay. Explain why arteries have thicker walls than veins. Okay. Because um the heart creates a big blood pressure for the blood to travel um to the body. Thus, the arteries have to be able to handle that blood pressure created. Thus, they have a large tunica media externa, and the wall is thicker than veins.
Good. That's
Wow.
that's
Okay.
2 marks. So you get the first one for talking about how they have to withstand the higher pressure from the heart to prevent them from rupturing or bursting. And you get the other point for saying that they have a thicker tunica media with more elastic fibers that
Yeah.
allows it to sort of withstand that blood pressure.
Yeah.
Okay, next question.
Short-term effects of each of these substances on the cardiovascular system. Okay. So carbon monoxide has a very high affinity for hemoglobin,
Mhm.
thus can replace the oxygen bound to the hemoglobin, and less oxygen is transported because of the carboxy hemoglobin created. It can also decrease or increase the amount of fatty acid layer, no, amount of the layer of fat in the arteries themselves, resulting in a stroke or CHD.
Okay, yeah. I'd leave out that last part just because the
Yeah, because it's not really short term.
yeah, exactly. Good. Well, talk to me about nicotine.
Okay, so nicotine, so when it enters the blood, it uh, um, travels to the brain where it can stimulate the nerves to release adrenaline from the um, adrenal glands.
Yeah, okay.
And it can stimulate.
Are the adrenal glands in the brain?
No.
No. Yeah.
And it can stimulate the brain to release dopamine, which makes it addictive.
So tell me what the effects of adrenaline are.
Increases the blood pressure and the blood rate. Sorry,
How does it increase blood pressure?
I have no idea.
So it causes the vessels, so mainly arterioles, to constrict.
Yeah.
That increases the resistance against which the heart is pumping, so that will increase the blood pressure.
Okay.
Nicotine also promotes clotting of the platelets.
Okay.
But again, it is a short-term effect. But in the long term, that would promote thrombosis and then the cardiovascular effects that you spoke about, such as stroke and myocardial infarction. Either carbon monoxide or nicotine also damage the endothelium,
Okay.
and that will like start the chain of events that will lead to thrombosis.
Yeah. Okay. Yep. Okay. So M equals I divided by A.
Yep.
Then you measure what I is.
I'm telling you, it's 30 millimeters.
Okay, so that's equal to 30,000 micrometers.
Yeah.
Then you divide 30,000 divided by 10, which should give you 3,000.
Good.
So just 3,000.
Good. You get 3 marks for that.
Okay.
So name structure X and state one function of the structure. Let's go with that.
Factorial.
Good. And one function.
Well, one function is that water travels through the symplastic way to the vacuole of plant cells.
Yeah, so it stores water.
Yeah.
Good.
Okay. So
Tell me
they're
about,
also
tell me
other
about
things.
other functions.
Huh?
Tell me other functions of the body.
Okay. So it contains. So it regulates the osmotic uptake of plant cells by regulating the water potential.
Yeah, good. So it sort of gives the plant cell turgidity.
Yeah.
Yeah. Other than water-related effects, anything else that the vacuole does?
Well, I know that it contains pigments and sugars.
Yeah, so the sugars is really important. So cell sap, it's a sort of store for the energy products that are being synthesized.
Yeah.
And it actually also stores the waste products.
Yeah.
Okay, so next question.
Okay.
So back up to this.
Nucleus and chloroplast.
Good. Any other organelle?
Mitochondria, but I can't really see it, I think.
Yeah, there are. Sorry, my mouse is kind of going crazy. There are mitochondria, but they're just smaller. So, like,
Is it towards the left bottom of the biggest? I don't know how to.
so up here,
Okay.
the sort of lighter shaded organelle. There are other ones as well, but
Oh,
they tend to be a bit smaller than the chloroplasts.
so I've got a question. So what are those? So the white spaces in the chloroplast, are
They-
they the grana or
Yeah, yeah. So these are the
that's what I thought. Okay. Okay. B is the capsid,
good.
and A is it's the nucleic acid. So it's the genetic information, aka the nucleic acid.
Yeah, good. And what could that nucleic acid be? What kind of molecule?
RNA could be RNA,
Could also be DNA.
DNA. Yeah, so it could be retrovirus, or it could just
Yeah, good. Okay.
okay. So DNA is transcribed, and the codes of so it's so the code of DNA is used to form codons. So the triplet codes of the gene of DNA are transcribed into codons of mRNA, and then the mRNA um, binds with like um, binds with specific tRNA molecules with specific anticodons, which are complementary to codons during translation to form specific um, polypeptides with a uh, specific amino acid sequence.
Yeah, good. So when you say that DNA, DNA, which is essentially coding for mRNA, in the context of the question, they're talking about a peptide called cysteamine.
Yeah.
So what part of DNA is important here? You have to.
Yeah. So we're talking. Yeah. Sorry. So we're talking about the nitrogen bases and the triplet codes that they create.
Yeah, and because we're talking about a single protein,
We're talking about a gene.
you have to mention that. If they ask you about the role of DNA in the production of protein, you have to mention that the gene of DNA. But yeah, go the other parts, right? So it's only a 2-mark question. So it's broad strokes here. Gene codes for the protein, and that gene is transcribed into mRNA. And
Yeah.
mRNA is translated into the protein. But remember, because they're asking about the role of DNA, don't need to talk about translation because that has no DNA.
Okay.
Okay, next question.
Okay. So protease enzymes in herbivores have a complementary active site to the inhibitor. Oh no, sorry. Okay. So serpin inhibits only the enzymes, the protease enzymes of the herbivores, and not those in plants, because the enzymes of the herbivores have a complementary active site to serpin, while those of plants do not. So no enzyme substrate complexes formed and turned into products.
Good. Okay. Next question.
Mm. Okay. So the presence of competitive inhibitors make it harder for the enzymes to reach maximum velocity.
What do you mean by harder?
Yeah, so by binding temporarily to the active site of the enzyme, they inhibit the substrate to bind to the active site. Thus, at the same substrate concentration, less of the substrate would be bound to the active site. Thus, Vmax is reached at a higher substrate concentration, and thus half of Vmax is reached at a higher substrate.
Good. That's good. Right. Let's take a break there. In 5 minutes, we'll come back.
Okay. Hello?
Hi.
Okay. Name the causative. Mycobacterium tuberculosis.
Good. Nice.
Deaths. Vaccine 19. And 1940. Okay.
Before you start, I'm just gonna put 2 arrows. 1, 2, 4. That's
Yeah.
good.
Okay. So when the vaccine is from the time that the vaccine is introduced, you can see a decline in both the mortality of tuberculosis and new cases of tuberculosis, followed by the curve plateauing, both of them.
Okay.
So does it say explain? Oh, okay. So by the vaccine being introduced, it means that more people weren't susceptible to tuberculosis disease, because they've created immunological memory against it.
Yeah.
So the secondary response rather than the primary response would be triggered.
Okay.
Okay.
So when I look at this graph, the first thing like that you want to comment on when they're doing quite a broad question. Sorry, I didn't show you. It's a 5-mark question, so
Okay.
they're going to expect quite a lot. I would just start with like the most simple points. So,
Okay. So there's for deaths from TB, there's a decrease of deaths from 1925 to 1980.
Good.
After that, it stays at 0.
Good.
And for the number of cases, there's an increase until 1940, I don't know, 7 or
Yeah.
and then a decrease, a general decrease. Yeah.
And so, not talking about how you'd write the answer now, but just ask you a question. What do you think has been affected more by the introduction of antibiotics and vaccines? The deaths from TB or the new cases?
For from the antibiotic, what's been affected?
Yeah, for both.
Yeah. So the number of cases hasn't been infected, but the number of deaths has. So treatment with the antibiotic was successful by treating already infected people,
Yeah.
but it didn't prevent other people from being infected and the cases of tuberculosis increasing.
Good. And the vaccine?
The vaccine was successful in both areas, so it decreased the number of deaths and the cases.
Good. And I think there's an argument for saying that actually looking at this data, we can distinguish between the effects of the antibiotics and the effects of the vaccine.
Could you repeat that, please?
So do you think it's possible here that we actually can't for certain say that the antibiotics is causing the decrease or the vaccine is causing the decrease? It could be
It
the
could be
fact that they're
delayed.
both.
Well, I don't think we can say for certain. I don't. Yeah.
Yeah, I mean, it's clear to me that there has been an effect following the introduction of these two treatments, but to say for certain how much either of the treatments contributes to the effect it's sort of all up in the air because the time period between the introduction of the two is so short. It could be that antibiotics alone would have eventually led to a decrease in the number of cases of TB and an elimination of the deaths if it was widespread enough to sort of eliminate all cases.
Yeah.
But we know that vaccinations will introduce herd immunity and sort of speeding up that whole process.
Yeah.
But you get marks in this question by commenting on things like the fact that the number of deaths was increasing from the start anyways before the introduction of the antibiotic and the vaccine. So perhaps other factors came into decreasing the number of deaths, for instance, sterile techniques in surgery or more hygienic practices that reduce the spread of the bacteria.
Yeah. So like external.
Yeah, to me, looking at that graph, to me, I'd say that the number of deaths from TB was decreasing anyways, and this just hastened that decrease.
Yeah. Okay.
And I mean, you could the number wasn't changed that steeply after the introduction of antibiotics, but it was more so the introduction of vaccines that increased the rate of decline in
Sure.
terms of deaths. Yeah. And there's a sort of plateau phase between the introduction of the vaccine compared to the deaths, which are immediately decreasing following the vaccine. The number of cases sort of plateaus before it starts to decrease. Do you know what that might be?
It plateaus before it starts to decrease the number of cases.
Yeah.
So that might be
That plateau.
because it takes a longer time to achieve herd immunity.
Exactly. That's it. That's exactly the point. Okay. And obviously, we won't go through it here, but in your exam, you're going to want to explain, you're going to want to describe the trends in quite some detail. So, you know, this line plateaus, then falls, or this line increases. You give the dates at which it happens, and give the Y value as well.
Yeah.
Okay, good.
What was the highest? Okay. So HIV positive people have a weaker immune system because the virus targets the T cells and destroys a lot of them, thus decreasing the concentration of T cells within the body over time. And this can lead to those people being immunosuppressed and more susceptible to diseases, thus causing an increased rate of mortality.
Yeah, and is it HIV that decreases the?
Well, it's AIDS.
Yeah, okay.
So it's HIV infection which leads to AIDS, and then AIDS which leads to
Good.
the
And
increase.
in your answer here, you want to specify that it's AIDS that weakens the immune system.
Yes.
So, infected at any given time would necessarily have a much weaker immune system than the general population. It's only the people in the late stage infection where that's
Yeah.
good. The fact that it's number of deaths from TB as opposed to number of cases, can you maybe comment on why is it that more people with HIV will die from TB?
So why it's numbers of deaths and not numbers of cases?
Not necessarily. I'm not necessarily asking you to make that distinction. Just tell me why people are more likely to specifically die from TB if they have HIV.
So sorry. So the question is why people are more likely to die from TB if they have HIV. Oh, okay. Okay. So having the HIV infection can lead to, as I said, decreased numbers of T cells,
Yeah.
which leads to so tuberculosis being either activated while it was inactive beforehand, or once infected with tuberculosis, it's already active
Yeah.
because of the weakened immune system.
Yeah, and what do you mean by active?
So by active, I mean that you so the person infected with tuberculosis actually gains, well, not gains, but has symptoms,
Yeah.
and yeah, it actually, yeah,
It causes the damage. So
yeah,
you go from having an asymptomatic individual who could have the bacteria anyways,
Yeah.
but it's lying dormant, to it being activated because of a reduced immune system, which leads to it just proliferating inside the individual.
Yeah.
And leading to a host of pathology that ends up with the patient dying. In this question, you could pick up marks from saying that people with HIV don't respond as well to treatment for life-threatening diseases because their immune system is so dampened.
okay,
You also mentioned the fact that a high proportion of deaths from HIV is due to tuberculosis,
yeah,
or you could think, if you're thinking outside of the box, they're talking about Swaziland. People have a high rate of HIV infection. It could be the case that people who have HIV in Swaziland are less likely to be able to get the treatment that would save them from dying.
okay,
So it could be that they don't have the money. So there could be a correlation between, the ability to actually pay for treatment and
Yeah.
HIV.
Yeah.
Move on.
okay. So cellulose does not contain phosphorus.
Good.
phospholipid does.
Yep.
ATP is found in plants.
Yep.
Cellulose is also found in plants. Hemoglobin is not. Phospholipids are found in plants.
Good.
ATP does not contain iron. Cellulose doesn't contain iron. Hemoglobin does contain iron. So phospholipids, yeah, do have a structural role. Cellulose does have a structural role. Hemoglobin doesn't have a structural role. And I guess ATP doesn't have a structural role itself.
Yeah, good.
But it can, like, cause metabolic reactions to form a substance which does have a structural role.
Yeah, but in
But
itself.
it itself doesn't.
Good. Okay. So talk me through this.
Okay, so this is basically a condensation reaction happening between two amino acids. So you'll end up with a covalent bond between the C and the N of the amino group and the carboxylic group. So which is the peptide bond, and the H bonded to the N, the OH bonded to the C, forming a water molecule.
Yeah, perfect. Would you like me to draw out, or are you happy with that?
I'm 100% happy with that.
Okay.
Okay, so they can be receptors for cell signaling.
Good. One more.
They can be also receptors for endocytosis. And they can be cell surface antigens to distinguish cell cells from foreign.
Good. Yeah. If you're talking about, if they're asking you to state two functions, I would only talk about receptors in one of them. So you could talk about receptors for signaling
Okay.
and receptors for endocytosis.
So would endocytosis not give me a mark there?
If you wrote, for instance, if you said receptor signaling, that would give you a mark.
Yeah.
And if you went on to say endocytosis or receptor for endocytosis, you wouldn't get the second mark because
Okay.
glycoproteins are involved in signaling endocytosis, but for the purposes of your course, they want you to focus on, other key rules of glycoproteins,
Sure.
such as adhesion, cell adhesion.
Or I could have even said cytoskeleton.
Yeah.
Okay.
Right. 5 mark. Describe the process of DNA replication.
Okay, so DNA is DNA is unwound. So it unwinds and unzips with the help of the enzyme helicase. And the hydrogen bonds between the complementary nucleus, the complementary bases are broken.
Good.
So the free nucleotides form hydrogen bonds with complementary complementary nitrogen-containing bases on both strands of the DNA. So the free nucleotides contain 2 extra phosphates, which activate them, and the DNA polymerase binds to the both of the strands and goes along both of them in one direction, from 3 to 5.
Yeah.
And catalyzes the bonds between the pentose sugars and the phosphate groups, creating the sugar phosphate backbone of the new DNA strands. The new DNA, strands, and you end up with 2 DNA molecules by semi-conservative replication, which contain both one new strand and old strand.
Good. Good. Excellent. And I mean, you get the 5 marks for that, definitely. But just to test your knowledge, DNA proofreading. Can you tell me a little bit about that?
Okay, so I know that. So in the G2 phase, the DNA polymerase goes over the DNA again just to make sure that all of the nucleotide sequences are correct, bonded to the correct bases.
Good. And what process occurs if there's a problem there? If you're an enzyme and you detect that the wrong base is bound? What?
A substitution mutation.
Okay. Yeah. I mean, it's not technically.
Well, not really. So just like a nitrogen containing base is like swapped out.
Yeah.
Repair, DNA repair.
Exactly. Good. So yeah, I mean, you're right. It could have been a substitution mutation that caused the error.
Yeah, but it's a repair. Yeah.
Yeah. Very good. Okay. Perfect. Next question.
Telomere.
Next question.
During DNA replication. Okay. Transversion event the newly synthesized strand of a purine unaware into 2 phosphate that could be used instead of cytosine in a transversion event. Okay, so cytosine is a pyrimidine, and it can be swapped out for adenine or guanine.
Good.
Okay. Transition event is when an incorrect purine is used or an incorrect pyrimidine is used. Suggest why transversion events are less likely to occur than transition. Because in a DNA molecule, a pyrimidine is paired up with a purine, and so in transversion events, a pyrimidine would be paired up with another pyrimidine and a purine with another purine. It's more likely for the wrong pyrimidine or purine to be to bond with another purine or pyrimidine.
Yeah. Good. That's one.
Is that one?
Yeah. So the fact that purines normally bond.
Okay. Maybe because they have a similar shape
Good.
and thus makes the enzyme it easier for the enzyme to not notice.
Yeah. Good. So this is
Because it has similar shape. Yeah.
good. And in terms of the differences in shape between purine and pyrimidine, what's the main difference?
It's the nitrogen number of nitrogen bases. So nitrogen rings. Sorry, number of nitrogen rings. So 2 for purines and 1 for pyrimidines.
Good.
Or
And
3.
if you had a transversion event, then so normally you have pyrimidines 23, so you have a total of 3 rings in one step of the ladder, if you like. If you have a transversion event where you have 2 purines bound to each other, 2 pyrimidines bound to each other, you have either 2 rings or 4 rings in that step. And would you agree with me that that would sort of deform the DNA helix by changing the width at one point?
So, the quality of the volume has like decreased. I don't know.
Oh, sorry.
Okay, now it's much better. I don't. Okay.
It's okay. Sorry, my mic is it's a measly thing. It might have just twisted or something.
Okay.
What I was just saying was, if you have a transversion event,
Mm-hmm.
you'd end up with a total of either 2 or 4 rings
Yeah.
in a rung of the ladder.
Yeah.
And because that would demorph the DNA double helix, it would, first of all, make it really obvious to the proofreading enzymes that there's a problem.
Okay.
It would correct it. And second of all, it would have further, it would cause further problems during replication when DNA polymerase is adding on bases. So the polymerase would probably correct that at that stage as well.
Okay.
Okay, good. Question. Sorry, I don't know if I just jumped. Yeah, there we go.
Okay. So mutations can alter the gene of the DNA for cell division,
Good, one more.
causing an oncogene, thus resulting in the cell constantly dividing uncontrollably
Good.
and passing on that mutation to the cells that it divides into. And those uncontrollably dividing cancer cells form an irregular mass of cells called a tumor.
Good. Good, you get the marks there. You could use some other detail, for instance, that a tumor suppressor gene could have been switched off.
Okay.
The disruption to the cell cycle could be a shortening of interphase, so you don't get the checkpoints that you need. You could say that the tumor itself, or the cells that lead to a tumor, don't respond to signals to stop dividing, and that they don't go through programmed cell death. But yeah, you get the marks anyways. Okay, so describe this one for me.
Okay, so the hydrogen bonds between 2 water molecules are in between the H of the hydrogen, of the water molecule and the O.
Yeah.
And there's supposed to be 2 hydrogen bonds, like per molecule.
Yeah.
So you would see another hydrogen bond in between, like the O and another and the H.
Yeah. Okay.
Okay.
And the fact that you get sort of increase here in the electrons and decrease here, which causes them to pair up in the cloud.
Yeah.
Okay, good. I mean, literally all you have to do is draw that and you'll get 3 marks.
Okay. You don't have to draw the dipoles or anything?
So you get 1 mark for drawing the water molecules with the correct bonding, another mark for drawing the hydrogen bond, and the last mark comes from showing the partial charges.
Okay, so you do have to. Yeah. That's
Yeah.
Okay. Oh, that's the D. Okay, I get it. Okay, so movement of water in the sound depends on the force of attraction between water molecules. Cohesion.
Good.
State the property of water that results as a cooling water. High latent heat of vaporization.
Good. That's awesome. And that's the paper done.
Oh, perfect timing.
So next week, would you like to do the same thing? Would you like to try a multiple choice paper as well? Or what are your thoughts?
I'd love to stick to paper twos.
Okay.
That's
Sure.
no
We'll just,
thing.
we'll just stick with more paper twos next week then.
Yeah.
As always, just like message me on Slack if you want to go through anything specific.
Yeah. So how many of the 2017 papers have we covered so far?
So
Or are there any that we haven't covered?
yeah, there are some that we have to go
Okay. Could we so the ones that we haven't covered, could we like leave them?
Yeah.
Because like I want to go through some 2017 papers which I haven't seen before the test.
Yeah.
That's okay.
Of course, that makes sense.
Okay.
So we'll just go back to like years further back. Then I can leave out 16 and 15 as well.
That would be great.
Okay.
Awesome.
All right. Speak to you next week then.
Okay. Thanks.
Cheers. Bye.